Have you ever wondered what makes you, you? Why do you have your mother's eyes and your father's smile? The answer lies within the intricate world of your genome. Imagine a vast library filled with instruction manuals that dictate everything about you, from the color of your hair to your predisposition to certain diseases. This remarkable library is your genome, the complete set of genetic instructions found within each of your cells. Like a comprehensive blueprint, your genome contains all the information needed to build and maintain your body. This genetic information is encoded within a molecule called deoxyribonucleic acid, or DNA for short. Think of DNA as a long, twisted ladder, with each rung representing a specific sequence of chemical building blocks called nucleotides. These nucleotides come in 4 different types: adenine, A, thymine, T, guanine, G, and cytosine, C. The specific order of these nucleotides forms the genetic code, which determines your unique traits. Each gene, a segment of DNA, contains the instructions for building a specific protein. Proteins are the workhorses of our cells, carrying out a wide range of functions, from transporting molecules to catalyzing chemical reactions. In essence, your genome is the master conductor of your life, orchestrating the complex symphony of processes that keep you alive and thriving. Let's delve deeper into the fascinating world of genes and non-coding DNA. Imagine our DNA molecule as a long, winding road. Along this road, you'll find bustling cities representing our genes. These genes, as we've learned, hold the blueprints for making proteins, the essential building blocks of life. However, not all of our DNA is made up of genes. In fact, a significant portion of our genome consists of non-coding DNA, often referred to as the dark matter of our genetic material. For many years, scientists believed that non-coding DNA served no purpose. However, recent research has revealed that this dark matter plays a crucial role in regulating gene activity. Think of non-coding DNA as the traffic lights and stop signs along our DNA highway. They control which genes are turned on or off, ensuring that proteins are produced at the right time and in the right amounts. Without these regulatory elements, our cells would be in a state of chaos, unable to function properly. Understanding the interplay between genes and non-coding DNA is crucial for unraveling the complexities of human health and disease. By studying these interactions, scientists can develop new treatments for genetic disorders and gain a deeper understanding of how our genes influence our lives. While DNA serves as the master blueprint of life, another key player exists in the intricate world of genetics: ribonucleic acid, or RNA. Think of RNA as DNA's messenger and interpreter. DNA, residing within the cell's nucleus, cannot directly instruct the protein-building factories called ribosomes, which are located outside the nucleus. This is where RNA steps in. RNA acts as a go-between, carrying genetic information from DNA to the ribosomes. This process begins with transcription, where a specific segment of DNA is copied into a messenger RNA-mRNA molecule. The mRNA molecule then travels out of the nucleus and into the cytoplasm, where it encounters ribosomes. Ribosomes read the mRNA sequence and use it as a template to assemble amino acids into proteins. This process, known as translation, ensures that the genetic information encoded within DNA is faithfully translated into functional proteins. In essence, DNA and RNA work together in a harmonious partnership, ensuring the smooth flow of genetic information from the blueprint to the building blocks of life. Where do we find these incredible libraries of genetic information within our cells? The answer depends on whether we're talking about eukaryotes or prokaryotes. Eukaryotic cells, like those found in plants and animals, are more complex and contain a membrane-bound nucleus. This nucleus serves as the protective chamber for the cell's DNA, organized into multiple linear chromosomes. Prokaryotic cells, on the other hand, are simpler and lack a true nucleus. Their DNA, typically a single circular chromosome, resides in a region called the nucleoid. This region isn't enclosed by a membrane, allowing the DNA to interact more directly with the cell's cytoplasm. Think of eukaryotic cells as well-organized libraries with their DNA neatly stored within the nucleus. Prokaryotic cells, in contrast, resemble more of an open-plan workspace with their DNA readily accessible within the cytoplasm. Despite their differences in organization, both eukaryotes and prokaryotes rely on their genomes to guide their growth, development, and reproduction. Section 5. Size matters, a genomic perspective. Just like libraries vary in size, so do genomes. The size of an organism's genome is measured in base pairs, the fundamental building blocks of DNA. Interestingly, there's no direct correlation between genome size and an organism's complexity. For instance, the human genome contains roughly 3 billion base pairs. The genome of the African lungfish, however, dwarfs ours with a staggering 130 billion base pairs. Even the tiny water flea boasts a genome larger than our own, with approximately 31,000 genes compared to our 20,000. These variations in genome size highlight the diversity of life on Earth and the complex evolutionary forces that have shaped genetic material over millions of years. Section 6. Organizing the Book of Life. Imagine trying to find a specific book in a library with billions of volumes and no organizational system. It would be an impossible task. Thankfully, our cells have evolved ingenious ways to organize their vast libraries of genetic information. In eukaryotic cells, DNA is tightly coiled and packaged into structures called chromosomes. These chromosomes resemble tightly wound threads, ensuring that DNA is compactly stored within the nucleus. This organization is crucial for cell division, as it allows for the accurate distribution of genetic material to daughter cells. Furthermore, DNA is associated with proteins called histones, forming a complex called chromatin. This chromatin structure plays a vital role in regulating gene expression, determining which genes are accessible for transcription and subsequent protein synthesis. The intricate organization of DNA within our cells highlights the remarkable efficiency and elegance of biological systems. Section 7. Decoding the genome, sequencing the secrets of life. Unlocking the secrets hidden within our genomes is no easy feat. However, thanks to groundbreaking advances in technology, scientists have developed powerful tools to decipher the language of life. One such tool is genome sequencing, a revolutionary technique that allows us to determine the precise order of nucleotides within an organism's DNA. The process involves breaking down DNA into smaller fragments, sequencing these fragments, and then using sophisticated computer algorithms to piece together the complete genome sequence. This information provides invaluable insights into an organism's genetic makeup, revealing its evolutionary history, susceptibility to diseases, and potential for future adaptations. Genome sequencing has revolutionized the fields of medicine, biology, and agriculture, paving the way for personalized medicine, targeted therapies, and the development of disease-resistant crops. Section 8. Genomic Revolution, Transforming Medicine, Biology, and Agriculture. The ability to sequence genomes has ushered in a new era of scientific discovery, with profound implications for human health, our understanding of life, and the future of food production. In medicine, genome sequencing is transforming the way we diagnose, treat, and prevent diseases. By comparing the genomes of healthy individuals to those with specific conditions, scientists can identify genetic variations associated with disease susceptibility. This knowledge allows for the development of targeted therapies that address the underlying genetic causes of diseases rather than simply treating their symptoms. In biology, genome sequencing has revolutionized our understanding of evolutionary relationships between species. By comparing the genomes of different organisms, scientists can trace their evolutionary lineages and gain insights into the mechanisms that drive biodiversity. This knowledge is crucial for conservation efforts and understanding the interconnectedness of life on Earth. In agriculture, genome sequencing is being used to develop crops that are more resistant to pests, diseases, and environmental stresses. By identifying genes associated with desirable traits such as yield, nutritional content, and drought tolerance, scientists can breed crops that are better suited to the challenges of a changing climate. Section 9. The future of genomes, unlocking mysteries and shaping our world. As we delve deeper into the world of genomes, we uncover a universe of possibilities for improving human health, protecting our planet, and advancing our understanding of life itself. The future holds immense promise for personalized medicine, where treatments are tailored to an individual's unique genetic makeup. Imagine a world where doctors can predict your risk of developing certain diseases and prescribe preventive measures or early interventions based on your genetic profile. Genomic technologies are also playing an increasingly important role in conservation efforts. By analyzing the genomes of endangered species, scientists can identify populations at risk and develop targeted conservation strategies. The journey into the world of genomes has just begun. As we continue to unravel the secrets encoded within our DNA, we unlock unprecedented opportunities to shape a healthier, more sustainable future for all.